Hello everyone. This series is going to be the last series I'm going to commit to. It's some important techniques in life science, particularly techniques related to biotechnology. So this series is going to be the last one because my net exam is approaching. It's 26th of this month. It's 26th of December 2023. The last part of this 2023 year. Later there will be new year, so I hope I would like to complete the series before that. So thank you for listening. Let's start. So first technique is marker assisted selection or MAS. So we will be development of population, QTLS mapping, QTLS validation, marker validation, and marker assisted selection. That's how we need to do this. So in this technique, linkages are sought between DNA markers and agronomically important traits such as resistance to pathogens, insects and nematodes, tolerance to abiotic stress, quality parameters, and quantitative traits. The essential requirements for marker-assisted selection is a plant breeding program. Our markers should co-segregate or be closely linked with the desired trait. And an efficient means of screening large populations for the molecular markers should be available. The screening technique should always have high reproducibility across laboratories, and it should be economical to use and be user friendly. Then several strategies have been developed that allow one to screen a large number of random unmapped molecular markers in a relatively short time. There are rapid AFLP, RFLP, microsatellites, etc. These methods rely on two principles to generate hundreds or even thousands of potentially polymorphic DNA segments and rapidly visualized from single preparations of DNA and use of genetic stocks to identify among these thousands of DNA fragments those few that are derived from a region adjacent to the targeted gene. To determine which of the amplified loci lie near the targeted gene, two strategies are proved effective, like nearly isogenic line strategy, bulk segregant analysis, BSA. The next, nearly isogenic line strategy. Nearly isogenic lines are created when a donor line is crossed to a recipient line, and the resulting F1 hybrid is then back crossed to the P2 recipient to produce the backcross 1 generation, and a single individual containing the dominant allele of a target gene with P1 is selected. Selection for the target gene is normally made on the basis of phenotype. This BC1 individual is again backcrossed to P2, and the cycle of backcross selection is prepared for a number of generations. In the BC7 generation, most, if not all, of the genome will be derived from P2 except for a small chromosomal segment containing the selected dominant allele, which is derived from P1. So lines homozygous for the target gene can be selected from the BC7F2 generation. The homozygous BC7F2 line is said to be clearly isogenic with the recipient parent P2. Then bulk segregation analysis, or BSA. It requires the generation of populations of bulked segregates bulks where P1 and P2 are hybridized. The F2 generation derived from the cross will segregate for alleles from both parents at all loci throughout the genome. If the F2 population is divided into two pools of contrasting individuals on the basis of screening at a single target locus, these two pools will differ in their allelic content only at loci contained in the chromosomal region close to the target gene. Bulk 1 individual selected for recessive phenotype will contain only P2 alleles near the target, while bulk 2 plants selected for dominant phenotype will contain alleles from both P1 and P2. Dominant homozygote and heterozygote, bulk 1 and bulk 2 will contain alleles from both P1 and P2 at loci and linked to the target. Marker-assisted selection has become a promising and potent approach for integrating biotechnology with conventional traditional breeding. The breeders' interest on molecular markers revolved around certain basic issues that have been illustrated below, like resistance breeding. At present, breeding for disease and pest resistance is conducted in the segregating populations derived from crossed or susceptible cultivars with resistant donors. These populations are then selected either under natural diseases or pest hotspots. Resistance genes will help in identifying plants carrying these genes simultaneously, almost without subjecting them to pathogen or insect attack in early generations. So, pyramiding of major or minor genes into cultivars for development of durable resistance or multiple resistance. Pathogens in insects are known to overcome resistance provided by single genes. Single gene resistance are fragile and often broken down easily. Therefore, breeders intend to accumulate several major and minor resistance genes into one cultivar to achieve durable resistance. Durability of resistance has been increased from developing multiliners and by pyramiding of resistance genes. Molecular markers for hybrid vigor using molecular markers on a set of diallel crosses along 8 allied parental lines, widely used in Chinese hybrid rice production program. High correlation has found between specific heterogeneity and mid-parent heterosis. Microscopic techniques. Biochemical and molecular analysis are frequently complemented by microscopic examination of tissues, cells, or organelle preparations. Examples range from relatively simple observations by eye to more complex experimental manipulations and measurements. There are two fundamentally different types of microscope, the light microscope and the electron microscope. So we were learning about microscopic techniques. Next topic. Okay. So there are two fundamentally different types of microscope, the light microscope and the electron microscope. Light microscope uses a series of glass lenses to focus light as it interacts with the specimen in order to form an image. Whereas electron microscopes use electromagnetic lenses to focus a beam of electrons. Light microscopes are able to magnify a maximum of approximately 1500 times, whereas electron microscopes are capable of magnifying to maximum of approximately 200,000 times. Resolution is the ability to distinguish between two closely spaced points in a specimen is a much more reliable estimate of a microscope's utility. In microscopes, the resolving power is inversely proportional to the distance between the two objects. The resolution R depends on the angular aperture. The resolving power of microscope formula is given by R is equal to 0.61 lambda by Na condenser plus Na objective, where Na is n sine theta. So Na is a numerical aperture, theta half the angle of alpha of the lens, which depends on the focal length and the diameter of the lens, and refractive index of the medium between the specimen and the lens, lambda wavelength of the light that is emerging from the object under consideration. The simplest form of light microscope consists of a single glass lens mounted on a metal frame, a magnifying glass. All modern light microscopes are made up of more than one glass lens in combinations. Okay, the main components of compound light microscope include light source that is focused at the specimen by a condenser lens, light that either passes through the specimen or refracted back from the specimen is focused by the objective lens into the eyepiece lens. The images are either viewed directly to eye by through the eyepiece, or it is more often projected onto a detector, for example a photographic film or more likely digital camera. Digitally captured images are displayed on the screen of a computer imaging system stored in a digital format and reproduced using digital methods. The condenser focuses the light and illuminates the specimen with parallel beams of light. So then the specimen stage is a mechanical device that is finely engineered to hold the specimen firmly in place. The stage enables the specimen to be moved and positioned in fine and smooth increment, both horizontally and transversely in the x and y directions for locating a region of interest. The Na is a measure of the ability of lens to collect light from the specimen. Lens with a low Na collect less light than those with a high Na. The resolution varies inversely with Na, which infers that higher Na objectives yield the best resolution. The visible spectrum, the spectrum of white light. Then contrast in the light microscope. Most cells and tissues are colorless and almost transparent and lack contrast when viewed in a light microscope. Therefore, to visualize any details of cellular components, it is necessary to induce contrast into the specimen. This is achieved either by optical means using a specific configuration of microscope components or by staining a specimen with a dye, or more usually using a combination of optical and staining methods. So, bright field microscopy. Contrast in bright field image is usually produced by the color of the specimen itself. Bright field is therefore used most often to collect images from pigmented tissues or histological sections or tissue culture cells that have been stained with colorful dyes. In bright field microscopy, the sample illumination is illuminated from below and observed from above, while white light in contrast in the sample is caused by absorbance of some of the transmitted light in dense areas of the sample. Then dark field microscopy is there. Dark field illumination produces images of brightly illuminated objects or on black background. So this technique has traditionally been used for viewing the outlines of objects in liquid media such as living spermatozoa, microorganisms, growing cells in tissue culture, or for a quick check of the status of biochemical preparation. For lower magnifications, a simple dark field setting on the condenser will be sufficient. For more critical dark field imaging at a higher magnification, a dark field condenser with a dark field objective lens will be required, used in observing unstained specimens. Condenser is specialized to block most light from the source, contains an annular patch stop which disperses the light in various directions, resulting in a cone of light. And phase contrast microscope. Phase contrast is used for viewing sustained cells growing in tissue culture or for testing cells and organelle preparations for lysis. This method images differences in refractive index of cellular structures. Light that passes through thicker parts of the cell is held up relative to the light that passes through thinner parts of the cytoplasm. It requires a specialized phase condenser and phase objective lenses. Each phase setting of the condenser lens is matched with the phase setting of the objective lens. Partly coherent illumination produced by the tungsten halogen lamp is directed through a collector lens and focused on a specialized annulus, like labeled lens or annulus, positioned in the substage condenser front focal plane. So wavelengths passing through the annulus illuminate the specimen and either pass through undeviated or are diffracted and refracted in phase by structures and phase gradients present in the specimen. Undeviated and distracted light collected by the object is segregated at the rear focus plane of the plane faceplate. Okay. Then differential interface contrast. It is a modification of phase contrast microscope. The DIC images of the two focal planes of a multicellular alga volvox. Then fluorescent microscopy is there, using fluorescence. And then laser scanning confocal microscopy. Measuring cellular dynamics. The electron microscope. TEM, transmission electron microscope. SEM, scanning electron microscope. Cryo-electron tomography. This is something new I have never heard before. New methods of fixation continue to be developed in an attempt to avoid the artifacts of specimen preparation and observe the specimen more closely to its living state. Specimens are rapidly frozen in milliseconds by high pressure freezing. Under these conditions, the biochemical state of the cell is more likely to be preserved. Many such frozen hydrated samples can be observed directly in the electron microscope, or they can be chemically fixed using freeze substitution methods. For this later method, fixators are fused into the preparation at low temperature, after which the specimen is slowly warmed to room temperature. So cells are rapidly frozen, fixed by freeze substitution, and embedded in epoxy resin. So thick 200 nm sections are cut and imaged in the TEM equipped with a tilting stage. A typical tilt stage is a series of 100 to images collected in a digital form and exported to a computer reconstruction program for analysis. Okay, electron tomography of a nuclear pore. The nuclear pore is a complex of three rings of proteins that allows the passage of materials in both directions between the nucleus and the cytoplasm, which we could see. Then, which microscope and contrast methodology might you use for the following applications? Fixed and fluorescently labeled zebrafish embryos, epifluorescence light microscopy, then sonicated cell extract to quickly assess the extent of cellular damage, light microscopy, dark field of phase contrast optics, then macromolecular structure of a virus, which we can only view through the electron microscope or electron microscope tomography, then screen GFP labeled gene expression in living fruit flies, which is with the help of fluorescence spectromicroscopy. Then fixed and stained sections of liver biopsy to check for cancer cells. Light microscopy, bright field optics. Surface structure of yeast cells. Scanning electron microscopy, because that's surface structure. Then macromolecular interaction in a living cell. Super resolution light microscopy. Then swimming spermatozoa 8. Dark field phase contrast, differential interface contrast light microscopy. Then colorful surface structures of a butterfly wing. You could only see that with spectromicroscopy. Then imaging deep into a living GEF labeled embryon developing zebrafish embryo over time. Multiple photon or light screen microscopy. And pedigree analysis. Generation 1 parents, generation 2 children, generation 3 grandchildren, male unaffected, male affected, male affected diseased, then female unaffected, female affected, female affected diseased. Disease in the sense dead, I guess. So a pedigree is a pictorial representation of a family history, essentially a family tree that outlines the inheritance of one or more characteristics. The affected individual through whom the pedigree is discovered is called the proband. Propositus if a male, proposita if a female. Proband, propositus if a male, proposita if a female. So unaffected person affected with trait, obligate character carrier or gene carries the genes but does not have a trait. And asymptomatic carriers, unaffected at this time but may later exhibit trait. Multiple persons, 5. Diseased person, then proband, first affected family member coming to attention of genetics. Family history of person unknown. Family, parents and 3 children, 1 boy and 2 girls in both birth order. Adoption, in brackets, close adopted persons. Dashed line denote adoptive parents. Solid line denotes biological parent. Dashed line denote adoptive parents, but brackets enclose adopted persons. Then solid line denotes biological parent. Then twins, identical, non-identical, unknown. Then consanguinity, mating between related persons. Double lines indicates consanguinity, consanguineous marriage. And then autosomal dominant trait. Autosomal dominant traits appear in both sexes with equal frequency, and both sexes are capable of transmitting these traits to their offspring. Every person with a dominant trait must have inherited alleles from at least one parent. Autosomal dominant trait therefore do not skip generations. Every generation will have one of them. So a trait that is usually considered to be autosomal dominant is familial hypercholesterolemia, an inherited disease in which blood cholesterol is greatly elevated owing to a defect in cholesterol transport. So cholesterol is transported through the blood in small soluble particles called lipoproteins. Principal lipoprotein in the transport of cholesterol is low density lipoprotein LDL. When an LDL molecule reaches a cell, it attaches to an LDL receptor which then moves the LDL through the cell membrane into the cytoplasm where it is broken down and its cholesterol is released for use by the cell. Then autosomal recessive traits. Autosomal recessive traits normally appear with equal frequency in both sexes unless penetrance differs in females and males, and appears only when a person initiates two alleles for the trait, one from each parent. So, autosomal recessive traits often appear in pedigree in which there have been consanguineous maintenance because these traits tend to skip generations. Autosomal recessive traits so appear only when both parents carry a copy of the gene for the trait, which is more likely when the parents are related. Autosomal recessive then usually arise in children born to parents who are unaffected and appear equally in males and females. A number of human metabolic diseases are inherited as autosomal recessive traits. Like one of them is Tay-Sachs disease. The disease results from the accumulation of a lipid called GM2 ganglioside in the brain. A normal component of brain cell, GM2 ganglioside, is usually broken down by the enzyme called hexosaminidase A, but children with Tay-Sachs disease lack this enzyme. Excessive GM2 ganglioside accumulates in the brain, causing swelling and ultimately neurological symptoms. Then X-linked dominant traits. X-linked dominant traits appear in males and females, although they often affect more females than males because it's kind of X-linked. So each person with the X-linked dominant trait must have an affected parent with X chromosome, most probably. So unless the person possesses a new mutation or the trait has reduced penetrance. X-linked dominant traits do not skip generations. Affected men pass the trait on to their daughters and none of their sons. In contrast, affected women, if heterozygous, pass the trait on to about one third of their sons and about one by two of their daughters. A male inherits the X-linked trait only from its mother. The trait is not passed from father to son. A female, on the other hand, inherits an X chromosome from both her mother and her father. So females can receive an X-linked trait from either parent, so their chances will be higher. An example of X-linked dominant trait in human is hypophosphatemia or familial vitamin D resistance rickets. Results from the defective transport of phosphate, especially in cells of kidneys. People with this disorder excrete large amount of phosphate in their urine. And X-linked recessive traits. X-linked recessive traits. First, these traits appear more frequently in males than in females because males need inherit only a single copy of allele to display the trait, whereas females must inherit two copies of the allele, one from each parent, to be affected. So second, because the male inherits 6 chromosomes from his mother, affected males are usually born to unaffected mothers who carry an allele for the trait. Because the trait is passed from unaffected female to affected male to unaffected female, it tends to skip generations. Which one? X-linked recessive traits. X-linked recessive trait skip generations. Okay. When a woman is heterozygous, approximately 1.5 of her sons will be affected and 1.2 of her daughters will be unaffected carriers. A third important characteristic of X-linked recessive trait is that they are not passed from father to son because a son inherits his father's Y chromosome, notice his X. So X-linked recessive trait will never pass from father to son. An example of X-linked recessive trait in human is hemophilia A, also called classic hemophilia. Results from abnormal or missing factor 7, one of the proteins in the clotic cascade. Bleed excessively, even small cuts and bruises can be life-threatening. Y-linked traits. Only males are affected. With the trait is passed from father to son. If a man is affected, all his male offspring will also be affected. Y-linked traits do not skip generations. So here you could see a comparison of four types, four or six types. Yeah. Autosomal recessive trait usually appears in both sexes with equal frequency. Tend to skip generations. First, let's learn autosomal dominant trait. Usually appears in both sexes with equal frequency. Both sexes transmit the trait to their offspring. Does not skip generations. Affected offspring must have an unaffected parent unless they possess a new mutation. When one parent is affected, heterozygous, and the other parent is unaffected, approximately half of the offspring will be affected. An autosomal recessive trait usually appears in both sexes with equal frequency, tends to skip generations. Then affected offspring are usually born to unaffected parents. When both parents are heterozygous, approximately 1/4 of the offspring will be affected. Autosomal recessive. Then X-linked dominant. Usually affected, both are affected, does not skip generations. Y-linked trait, only males are affected, passed from father to all sons. X-linked recessive trait. Usually more males than females are affected. Affected sons are usually born to unaffected mothers. Thus, the trait skips generations. Autosomal dominant hypercholesterolemia, autosomal dominant Huntington's disease, autosomal recessive sickle cell anemia, and autosomal recessive cystic fibrosis. Another one, Tay Sachs disease. So, hypercholesterolemia and Huntington's disease both are autosomal dominant. Sickle cell anemia and cystic fibrosis, along with Tay Sachs diseases, autosomal recessive. Then X-linked recessive, Lesch-Nahan syndrome. That's a deficiency of HGPRT enzyme. Then X-linked recessive, color blindness, insensitivity to light. Then X-linked recessive, muscular dystrophy, deficiency of protein dystrophy. The Lodescore method for analyzing linkage of human genes, mapping using somatic cell hybrids. How to do this? Linkage mapping using molecular markers, restriction fragment length polymorphisms. The distance between two linked RFLPs can be determined. Linkage mapping commonly uses molecular markers called microsatellites. A complementation test can reveal if mutations are in the same gene or in different genes. Genetic mapping methods. The concept of genetic mapping with 2-point test crosses, gene mapping with 3-point test crosses, then establishing the order of genes, calculating the recombination frequencies for genes, inference and coincidence, then genetic mapping in haploid eukaryotes. Ordered tetrad analysis can be used to map the distance between a gene and the centromere. Unordered tetrad analysis can be used to map genes in hybrid crosses. If no crossing over occurs in the region between the two genes, the parent type will be produced. And Mendelian genetics. Probability. The binomial expansion equation can be used to predict the probability of an unordered combination of events. Then deviation from Mendelian principles. Multiple allelism, ABO blood groups, for example. Then incomplete dominance in case of Mirabilis jalapa. Penetrance, expressivity, constant expressivity, identical non-genotypes with non-expressive effect yield 100% affected phenotype, variable expressivity, sex-linked inheritance in case of Drosophila, and sex-limited traits, sex-influenced traits. The sex Limited rates. In some cases, the phenotypic expression of a genotype is influenced by the hormones present in that individual, thus the trait is only expressed in sex-limited rates, usually determined by autosomal genes concerned to the development of a secondary sexual characteristics. Sex-influenced rates. Hormones affect less the alleles of the gene such that traits appear in both, but the alleles are dominant in one and recessive in opposite. Pattern blindness is an example. Pattern baldness. Pattern baldness is controlled by an autosomal gene with an allele of baldness. It's dominant for males and recessive for females. Pattern baldness. So baldness appears to be related to the level of the male sex hormone. So homozygous dominant genotype in males and females cause baldness. So heterozygous male is bald but female is not bald. BB man bald. Woman bald. BB bald in case of man. Non bald in women because their hormone level is not that much high. BB non bald. I hope this understands.